So, dear colleagues, welcome to session 2 of this SFB. It's on imaging targets and the probes, and we heard this morning by Elisabeth de Vries how important it is to define, to characterize and validate targets before synthesizing probes. And I'm glad Bernd Pichler made it to Munich after surviving a hailstorm last night, and he will give the introductory lecture on, and the title is Next Generation Imaging Targets, and I trust that everybody knows Bernd here, so no need to introduce him. Bernd, please.
Thank you very much, Helmut, for this kind introduction. Thank you very much, Markus, for inviting me. It's really great always to be back in Munich, where at least I have been exposed for the first time to imaging 25 years ago, and where my career started together with you, with Sybil, Wolfgang, the Girls Club of Professor Senikovic, which are also on the back, and the three rats, Haubi, Schlaubi, and Traubi, what they kept as pets, leftover rats. And I heard one we know is one radiochemist, and Wolfgang, the other one, were renamed. Haubi was Roland, Traubi and Schlaubi, and we can think who is what, who. So anyway, it's really always great to be back, and I'm really happy to give this lecture. So next generation of imaging targets, and I would like to explain a little bit what we think in Tübingen and what we build our program of about next generation of imaging target. And we group them in 4 different areas. One is the metabolism, like FDG, FET, FLT, and F-MISO. The other one we grouped in the tumor microenvironment, what we think is very important also to access more in detail. And also we are very focusing on tumor stress and tumor stress responses, as well as we are highly interested in the immune microenvironment. However, it's not a question of whether the one or the other is more important. It's really the convolution of all of those, and we need all of them. So I would say it's converging of imaging biomarkers and deconvoluting then of the respective signals. And when I say converging of imaging biomarkers of tracers, it means also that we need converge and bring together the different modalities. And therefore, what Simon showed, it's essential for our studies, and I will show you this later, why we really need higher detection sensitivity, shorter scan time, that we can scan more often and profile a tumor during the growth and guide tumor therapies. And the other thing is what I liked from Van Schilling, who showed really the importance of MR signals, because I really still think that the combination of different signals from MRI and PET are really essential. So I would like to briefly go in my talk on examples of all 4 of those areas. And the first one is what we are highly interested, imaging of tumor stress and specifically senescence. And therefore we built a tracer, and I will show you this in more detail just in a minute, the FPY-GAL tracer, which is a senescence tracer. The uptake we have found out is through the GLUT transporters. And this is one of the tumor stress, which I will explain in detail. Another important factor for tumor stress is the replicative stress. And therefore we use the PARP tracer, and not the same way as Susanne uses the PARP tracer, but we really want to use the PARP tracer for a replicative stress of tumor cells. So what is tumor senescence? Tumor senescence is a durable cell cycle arrest. It's induced either by DNR damage or other stress factors. So you can use radiation therapy or drugs, chemotherapeutics, to induce tumor senescence. It's important really to diagnosis, and it's important for therapy monitoring and for guidance of therapies to know about the state of senescence. It is linked to growth arrest, metastasis, therapy resistance, and you have the option to use senolytics to get rid of these senescent cells. So therefore, our aim was to develop a PET tracer that enriches in senescent tissues and senescent cells for non-invasive imaging, and therefore we used beta-gal compound. So again, what is senescence? Senescence plays a specific role in solid tumors. So if you have cancer and you treat the cancer, some of the cells might go into apoptosis or necrosis. However, this is the main process in non-solid tumors. In solid tumors, there's more and more understanding that a majority of cells go into senescence, which is an irreversible cell cycle arrest. So there's no proliferation anymore, but there's still glucose metabolism. Therefore, there were first attempts already a few years ago where FLT and FDG has been used. However, this is a little bit tricky. We would really like to have an imaging probe which is more specific for senescent cells. So the chromatin structure changes, and there is a higher expression of beta-galactosidase. What is the tricky part of senescence? So once you have cancer cells driven into senescence, they are of a secretory phenotype. So they secrete factors which do multiple actions. One, they do immune suppression, and therefore usually you have a hard time to treat the tumor, the remaining tumor, the viable tumor, which is not in senescence, efficiently. On the other hand, they trigger immune clearance to take away the senescent cells. However, there's an imbalance, and in the worst case, the tumor grows more by stimulating non-senescent cells, and therefore the tumor explodes. Ideally, it would go in a rest where you bring a tumor from an acute to a chronic disease. So what you can do is senolytic therapies, and there is, for example, navitoclax, which can be used to carry away and lyse these senescent tumor cells. However, to know the exact time points when you have the maximum of senescence, when do you still have to initiate senescence, or the time point when you have to take away the senescent cell, that's important to know. And actually, this is a field where imaging, what at least we think, can have a huge contribution. So what is the problem? The senescence trigger is usually, as I mentioned, DNA damage, and then there is a complex initiation of senescence programs where P16, P53, and P21 is involved. So there is not yet one specific senescence tracer. However, in the next step, there is this beta-gal induction, and you have this enzyme available within the cells. And that's exactly where we go to, because so far you can detect senescence only by immunohistochemistry, Western blot, or beta-gal staining, and ideally you need really the combination of all those ex vivo immunohistochemistry and histology probes. So we started with a known beta-gal substrate, and we radio-labeled it with fluorine 18, and this beta-gal substrate is taken up by GLUT1, and then it's cleaved and the radioactive part remains within the lysosome. That's the principle of this senescence tracer. This beta-gal substrate, what we have now, we got the full approval process, so we have in German the Herstellungserlaubnis, and we started now a full phase 1-2 study where we started like 3 months ago to include healthy volunteers for dose escalation, and then hopefully we have by end of the year also, or next year, the phase 2 study where we can also get biopsies finalized. However, we did a complete testing panel. We looked at the serum of mice, rats, and humans, whether the trace is stable. Also, we looked in HPLC when we cleaved it with beta-gal enzyme, the tracer in vitro, how the radiometabolite versus the non-radiometabolite look in the HPLC. We did extensive in vitro cell studies with gamma counting and incubation of cells. We had different cell lines where we did different treatments to elicit the senescence, like doxycycline removal for P53 reactivation, doxorubicin, or RCI. So really that we wanted to see whether all different therapy options to drive a tumor into senescence can really work with our tracers. So this is one example in an HCC, hepatocellular carcinoma mouse model. And you see over here the control, which has no senescence, so it was not treated to initiate the senescence. And this is actually the treated mouse, which goes into senescence, where the tumor goes into senescence, and as well as other tissue also. The autoradiography shows very clearly that there are clusters of senescence part within the tumor, so it's a heterogeneous setup, which means also that there are always non-senescent cells, and that's the importance that we can use imaging to detect the degree of senescence, and that we can use senolytic therapies to really delete the senescent cells. Also, this is the positive control, the beta-gal staining. You see over here the time activity curves for the control and the senescence tumors. We did this also on different other tumor models with different induction of senescence, and you see for the senescence versus the non-senescence mouse, we get usually, but keep in mind, it's always a heterogeneity of senescent cells and senescence clusters. Therefore, we need really a cluster analysis to get a single mouse analyzed. What we did then, after all these preclinical studies went very well, however, we got also several surprises during those studies. For example, that mice have an enzyme, apparently, which cleave the tracer, so therefore we used now rats, and there the tracer is very stable. We did also a compassionate use study, where we had, in this, for example, with the approved drug, a compassionate use study with a glioblastoma patient, and I will walk you through this patient scans. So we started in November 2018 with an FET scan, and had the diagnosis of this glioblastoma patient before we initiated senescence induction by the radiation therapy. Then we did the second scan 2 months later in January 2019, and you can see that the uptake of FET has really is really highly reduced. Especially over here in this part of the tumor, there is only a faint tracer uptake remaining. A day later, we did this senescence tracer scan with this FPY-GAL tracer. And surprisingly, what we found, also the FET uptake is much higher in this part of the tumor. The senescence tracer uptake was higher over here in this part of the tumor. So there was a clear mismatch between FET and senescence tracer uptake. Therefore, we went a step further with this patient and used a senolytic therapy. And what we could show actually with the senescence tracer, that the uptake was really removed dramatically after the senolytic therapy. So this was the first really study or trial what we used in a compassionate use, where we had one patient which were scanned by PET and MRI, FET, and our senescence tracer, and where senescence initiating therapy was applied as well as a senolytic therapy. So really the parameters came together, and we think at least that this is an interesting concept to image cellular stress and also adapt then the therapies. As I showed you that senescence is somehow comes together with an immune invasion, and the immune system plays an important role. And therefore, we are not only interested in one imaging factor, however, we are also interested to image the immune system. Our target cells are CD8 T cells and CD4 T cells, so interferon gamma producing T cells, as well as CAR T cells, but also PD-L1 and PD-1. The problem is really what is the right format of the biological you use, and this is a review I did together with Jan Grin and Fabian Kiesling last year for JNM, and we put together what is the circumstances of cells to small molecules and the different biologicals according to their size in uptake time and so on. And the problem is that the larger the biological is, the longer the blood circulation is usually. However, if you use a too small fragment, then it might clear too early and it might not really enrich in the tumor. For example, with FAP and SCFV fragments, we never had really success of our imaging approaches. Where we found very nice results was with nanobodies, what we got from alpacas, but also with mini-bodies or full-size antibodies. And I will show you some example what we did on imaging with different of these formats and also radio-labeled isotopes later on. Our main interest is really to image immunotherapy, and we have three main parts what we would like to image. One is the checkpoint inhibitors and T-cell engagers. Another important area is the vaccination, and our immunologist Hans-Georg Ramsey is working on DC vaccination and peptide vaccination. So he's really to image those immunotherapies. And the third part is the CAR T-cell therapy, where we use, for example, adapter CAR T-cell and radio-label those T-cells over biotin or with PSMA. So the goal of all these immune imaging is really to assess the T-cell response quantitatively and look on the T-cell infiltration, whether we have cold or hot tumors, as we heard already this morning from Liz. But also the T-cell migration and activation is extremely important, and I would like to go more further into those two areas, and also therapy-resistant mechanism. So we have established the CD8 mini-body from Imagineb just recently in Tübingen. We got also the manufacturing license, so I have the approval and also ship it to other centers. But we have also clinical studies running with this CD8 mini-body from Imagineb in our center. We want to understand better what the CD8 mini-body is really doing, because when you have the patient scan, it does not always look obvious what the enrichment is, at least in our data. So this is one example of a patient. This is one and the same patient with two tumor lesions, and you see over here there is an uptake, hot lesion, but the other lesion there is nearly no tracer uptake. So what we were interested, and therefore we used mouse studies with MC38 tumors, which is a partial responder tumor model, where part of them were responders, part non-responders. And then we used the FTY 720 drugs, which is an immunosuppressive drug that actually prevents the cells from migration. So therefore we simply stop the cell migration into the tumor. What we did, we injected the radio-labeled CD8 mini-body, and then we watched over time with only one injection over 7 days the cell migration. And if you look on this graphic, the non-responder and the model where we stopped actually the cell migration by this drug got the same curve, while there is an infiltration into the tumor tissue in the non-treated, or in only the PD-1 treated mice, but not in the non-responder and not in the mice where we stopped the migration by FTY 720. So therefore we believe that this is an important factor, that we not only look on a static situation, what is the uptake in a tumor, but also look on the temporal dynamics of the cell migration and see what migrates of the cells over the time into the tumor. And therefore, longitudinal scans, repetitive scans are extremely important, either with one or with multiple tracer injection. And this is actually our main interest, and we get hopefully in 4 to 8 weeks this total body PET scanner, what Simon showed, that we can follow these patients multiple time, have short scan times, or have low activity levels if we want to do multiple injection and scan them over several weeks or months. Another factor which is very important to us is not only CD8 positive T cells, but also the CD4 positive T cells are extremely important. So these are Th1 cells which express actually interferon gamma, and it was shown that interferon gamma plays an important role in PD-1 and PD-L1 treatment, but also in all the CAR T cell related treatments. And therefore we are interested to have the convolution of CD8 and CD4 T cells. And what we can see actually in this mouse model, which has not many CD4 T cells, that we get really a quantitative signal by using the CD4 radio-labeled minibody. One thing is immune cell infiltration migration. However, there is more than just having immune cells there. We are also interested whether they are activated or not. And there is one marker, which is the CD69, which shows the T cell activation. Therefore, we also radio-labeled this CD69 full antibody and with copper-64 and looked at the T cell activation. And actually, this is the convolution of CD3 T cells, so CD4 and CD8 T cells. When we stimulate them, we had definitely a higher binding than with non-stimulated. And the interesting part is shown over here when we have a T cell depleted mouse model and a mouse model without a therapy. We see really a high significant relation whether the T cells are activated or not. More importantly, what this slide shows over here in the longitudinal study, when we use the CD69 antibody for T cell activation, that we can distinguish already 3 days after therapy start whether this is a responder or non-responder. So this CD69 for T cell activation, what we believe and what we see in this animal model, allows us a very early prediction whether the mouse respond to the PD-L1 and LAG3, in this case, therapy or not. And actually, this is shown over here. If we have an over B16 melanoma model, which is a non-responsive model, then actually we don't have a significant difference between the treated and the control, but between the control and the over. So just the external transferred T cells show really an activation, but not the endogenous T cell. So this gives us a tool in our hand where we can very early predict, already 3 days after therapy start, whether immune checkpoint inhibitor therapy is successful or not. And now we want to go one step further with these methods and use machine learning. And in this case, it's a simple approach, a first simple approach by Gaussian mixture model analysis, where we want to cluster the uptake within the tumor into CD69 low and CD69 high. So we have therefore 3 different clusters with this Gaussian mixture model and use thresholds that we can really distinguish between a low and a high uptake of CD69 tracer and therefore T cell activation. We used with those tumors, based on an image-guided biopsy, then CyTOF and looked on the convolution of the immune cells within the tumors, and we could clearly distinguish the CD16, based on the CD69 expression, a complete distinguished immune cell setup in the different tumors. So therefore, using now machine learning, or in this case, an easy model of Gaussian mixture applying, then we can already distinguish non-responder and responder of tumor therapies by differentiating CD69 low and CD69 high. So the idea is really to go from imaging to in vitro data analysis by CyTOF and merge them together and retrospectively and iteratively learn for our imaging signals, more or less make imaging more precise. Now I would like to go in another example, which is the imaging of GD2. And this is more the microenvironment of the tumor. GD2 is overexpressed in neuroblastoma, so it plays an important role in neuroblastoma, and we also used mouse model with neuroblastoma. We radio-labeled a GD2 antibody, which is now approved for therapy of neuroblastoma, and we radio-labeled it with copper-64. We did a different mouse model, positive tumor cell line, a control, and using a control antibody, and we have significant higher uptake in the neuroblastoma with the GD2 and a radio-labeled antibody compared to the control cells or to the tumor what we exposed to a control antibody. So therefore we could, what you see also in the PET images, see the temporal uptake of the tracer in the animal model, but also see over here in the positive tumor, in the control tumor, or in the control antibody that there is a highly distinguished tumor uptake. We used now this copper-64 labeled GD2 antibody and injected it in young patients to guide this GD2 therapy. And what you see actually over here is a high uptake in this tumor lesion. Then the patient, it was a 14-year-old boy, underwent an 8-over-8-week GD2 monoclonal antibody therapy. And what we saw that actually this lesion went away while a new lesion came up. And we compared this with the MABG scintigraphy. And this is approximately the same what Liz de Vries said this morning. Those lesions which show an uptake usually respond to the treatment with an antibody, and those lesions which don't show an uptake at this time also don't respond to the treatment. Actually, we are currently looking into this, and I will show you in the next slide this. However, before I go more into the problems with antibody imaging, we just had recently, and this was a little bit surprising to us, how well it worked, a sarcoma patient with several metastases, and actually this was a patient from here, from Munich, which was TranslaTUM from the Haunerschen Kinderhospital, and we used our GD2 tracer, and actually they did now really based on the imaging information the GD2 monoclonal antibody therapy, and we will have this patient next month again in Tübingen and scan the follow-up. But for sarcoma, I think this looks really very promising that we can use an image-guided therapy with this monoclonal antibody. So what is the problem? I guess the problem what we have to learn with the antibody, therefore multimodality imaging is essential. Whether we have either a pre-dosing problem so that we need a cold antibody to block all other non-specific binding sites and that get therefore a more homogeneous distribution. It might also be that the target is not really positive for our tracer, so the receptor is not expressed, or whether we need a preloading, for example, with a non-specific antibody. All this, I guess, is better combined when we use multimodality imaging to get morphology, functional information like pH, like diffusion, like perfusion from the MRI, along with the antibody uptake from PET. So in my last few slides, I would just like to make a little bit of vision where we think it should go to, which is definitely going multimodal. So what we need is really high detection sensitivity and different information from different imaging modalities. But I would say we need more than imaging. We need also other modalities to really complement our imaging information and make it more precise. This is actually something what we saw today already, that we imaged the pH with CEST and MRI. And actually Andre Martins in our department established for our 7 Tesla MRI scanner in mice the acetocest MRI, where he can show, as we saw from Van Schilling this morning, what is the pH in a specific tumor. So why is this important? And Van showed today already a few examples. What we wanted to study is really the relation between acidosis interferon gamma, which is expressed by the CD4 positive T cells, and the PD-L1 expression. And what we found just recently, and the paper is currently under review, is what was known that acidosis somehow influences the PD-L1 expression. However, what we found now that it goes over an interferon gamma acidosis mediated and interferon gamma induced PD-L1 expression over the STAT phosphorylation. So therefore, this is an essential pathway, and we can, by using both acidosis imaging, but also manipulating the pH of a tumor tissue, we can influence the success or non-success of an immune checkpoint therapy. And therefore, we think this is extremely important to have really these different parameters together. What we also use with these different parameters, that what we get especially from PET MRI, the different MRI contrasts, the PET dynamic information, that we use stress, that we use this for distinguish stress factors like apoptosis, necrosis, fibrosis. And also this, what we usually do is that we train the data for these machine learning networks in an animal model and then translate it to the patient situation. And we did this now for 2 tumor entities. One is a colorectal cancer model in mice, and then we used the developed algorithms in patients, and one was on breast tumors. And actually it went really very well that we could establish the machine learning when we need a lot of data, many mice in a preclinical setup, and then translate it to the clinical setup, all the trained algorithms with these machine learning approaches. So therefore, preclinical imaging and clinical imaging really comes together by training those algorithms for machine learning and artificial intelligence. The last point I want to just briefly show in 1 or 2 slides is that we also look into intratumoral heterogeneity by metabolomics, proteomics, and genomics. And therefore, we build a milling machine where we can scan rats or mice in PET, MRI, and CT, then freeze them in liquid nitrogen. This goes within 2 minutes, and then have it in a milling setup. And you see over here the milling drill bit, and then get really by an accuracy of better than 0.1 millimeter tumor samples, what we can use then for in vitro analysis of metabolomics, proteomics, and genomics, and then relate this to our imaging results. So therefore, we don't want to have only the different metabolic tracers and use metabolic information by imaging, use also machine learning, but also we want to use the in vitro information by mapping the information of a specific imaging profile to metabolomics and proteomics information. What we want to also use is metabolic inhibitors, that we inhibit specific metabolic pathways and have then a deconvoluted information by machine learning of just one metabolic factor at a time. And the last thing I would like to show where we are working on, and this is Andre Martin's focus, that we do also metallomics, that we use different metal markers in PET and MRI and see how they can map the tumor heterogeneity and what information those different metals like zinc, copper, calcium, or manganese can deliver as additional information to our PET tracers and MRI signals. However, we need also the multi-scale analysis, and therefore we have now a full setup of intervital microscopy by Bettina Weigelin, and also we want to learn on a cellular basis the immunomicroenvironment and then map it back to the macroscopic scale and hopefully understand better the immunomicroenvironment and improve the immunotherapies. So bringing all this together, the immune therapy, the combinational therapy, and the molecular targeted therapies, we need also to image those different factors. We get a lot of different data from different origins, and I guess we need machine learning and artificial intelligence to bring those factors together and finally get the clinical relevant information out of it. And this is actually the study setup we want to use in several clinical studies, that we have a basic imaging of metabolomic and anatomy, like PET MRI or PET CT, the disease-specific immune imaging with the dynamic PET, with all the MRI information from the different MR protocols, that we use the multiparametric heterogeneity tumor profile by machine learning, and then that we do a longitudinal scan protocol, that we pick the relevant imaging parameters, that we start the treatment and we do a longitudinal imaging, and then use again machine learning for longitudinal multiparametric imaging information, and either it's a responder and therapy continuation or a therapy adaptation where everything starts from front. So Simon, it would be great to have the high sensitivity PET CT along with an MRI. Maybe the next project for UC Davis. With this, I would like to thank my group, and especially also the clinical collaboration partners, specifically from the nuclear medicine, Christian Lafauchère, but also Lars Zender, our oncologist, where I do all the senescence and tumor stress project, Costa Nicolau, radiology, Letizia, the pathology, and our oncologist and immunologist, as well as the senior group leader in my department. Also the funding, especially the IFID Cluster of Excellence, which funds most of the work. And last but not least, I would like to thank you for your attention. Thank you very much.
Thank you very much, Bernd, for your visions and your views into the future. Are there questions from the audience, please?
Yes, it was a great talk. Thank you very much for that. With the implications actually of immunotargeting of T cells, you chose different surface markers, subpopulations for activation. I mean, they are important for signaling as well as chatting between immune cells. So what do you think is the impact of the imaging on those actually markers then on their function? And what do you think is our task for translation or regulatory work to bring them into the clinics?
That's a very good thing, and you know it's very complicated. I think what we have to distinguish it is really between a static environment and a dynamic. That's why we are interested in this. Not only is there an enrichment, but is there an activation, and is there still a penetration over time? So the temporal effects are extremely important. And I guess saying this, that the question will be, do we get enough information to really deconvolute those factors you just named by, so for example, one single injection, or do we need multiple tracer injection? And I guess, honestly, my opinion is this anti-immune imaging is so important for the imaging community. If we get this to fly, I think we are done. And the reason is the therapies are so expensive. I mean, a checkpoint inhibitor therapy, I guess it's still about 100,000 euros. CAR T cells of 400,000 euros. So the imaging costs are marginal. But I guess what we have to learn is what tracer do we need, what imaging markers do we need, and is it, for example, one injection, is it multiple injection? And therefore, I really look forward to this high sensitivity scanner, because I think with the doses we apply currently, we have no chance. Then we can do a one-time imaging, especially for the patients who get cured, which are hopefully a lot with those immunotherapies. But I think we need to get really the dose down. And that's the main motivation, and Simon might have many more motivations for this high sensitivity scanner, but the dose reduction for immunotherapies is the main motivation for us. And I think we have to learn this, how this works really. But I cannot answer straight away your question, because that's what we have to really find out in the next years.
I think we are running out of time, and have to...
Thank you very much.
Thank you, Bernd, again. So now we have 4 talks of different projects. And again, I assume that you all know each other, so I don't have to introduce you. And the first talk is from Natalia Pellegato, and it's Project B8.
Yes, so thank you for the invitation to present some of the data that we have accumulated over these 3 funding periods. So as you can see, the focus is actually a preclinical model. And the aim of the first funding period was to characterize this preclinical model with a specific focus on imaging and see how we could use this model for translational research, especially going into novel therapies for neuroendocrine tumors. And actually, this model turned out to be really a gold mine of information. First, it told us about a new gene that is involved in the genetic predisposition to multiple neuroendocrine tumors. And then within this SFB, and all the characterization that we did at the molecular level, we were able to actually better understand the molecular pathogenesis of some neuroendocrine tumors, the ones that I will present to you later on. And based on this information, we identified also novel pathways involved in these tumors, novel therapeutic targets, and we could validate or evaluate the potential of these therapeutic targets in some preclinical studies in vivo in collaboration with other members of the SFB. So this is just to illustrate the phenotype of these animals. So this is, these animals are affected by multiple endocrine tumors, and I won't talk about the genetics, so the defect that causes these tumors, but what is important is that this was a spontaneous tumor model, so that there was no genetic modification. So this mutation in the causative gene, which is P27, occurred spontaneously. And what is important is that this is an endogenous model, so the tumors develop within the microenvironment where they usually develop. And we see also a progression from early stages to late stages within a few months. So by 8 or 9 months of age, our animals develop with full penetrance the tumors that we'll talk to you about. And this is also an advantage when you plan in vivo studies with an endogenous model, that we are sure that that our rats actually have the tumors that we want to study. So what are the clinical issues associated with NETs? So I simplify a lot. So based on the new WHO classification from 2017, all NETs have the potential to become malignant, and therefore most of them will be incurable. So the first question is how can we predict which tumors will become malignant? So there is the need to develop new biomarkers. Then another important question is which treatments can work for these tumors when they become unresectable, aggressive, malignant. And so there is a need for novel therapeutic targets. And then we wonder, okay, can we give some contribution to try to address these issues with our animal model? And what I will show you today is mostly related to this second question about novel therapies for these tumors. So the strategy that we followed over the years was to really characterize the tumors of these rats at the molecular level, and then validate these findings in human tumors. Then select from these novel pathways or genes that we would find the ones that could be druggable. And then go back to some in vitro drug testing using also the primary tumor cells from our animals. And then eventually, if this was promising, then going back to some preclinical in vivo studies, and imaging, of course, helped us. It was actually instrumental to monitor therapy response in vivo over time. And this, of course, is in view of this of a potential contribution to novel therapies for patients. So this is, as I said, so these rats develop multiple tumors, and today I will just focus on 2 of them. So we started our studies on these adrenal tumors, these pheochromocytomas that arise from the adrenal medulla. And I will show you data basically from a few studies. They were mainly led by Hermine Moore, a postdoc in the lab who was sponsored by the SFB 824, and Sebastian Gulde, a PhD student in the lab. So recently, as I said, one big part of our work was first to characterize the tumors developing in this animal model in order to figure out how or which kind of tumors they will mainly resemble, if they could be used to model the human tumors. And we recently were able to, by characterizing at different levels these tumors, we could show that they actually they share features with pseudoepoxic pheochromocytomas that in patients are the most aggressive tumor subtype. So this information will lead further studies then where we can really guide our therapies that we want to evaluate in this tumor model based on this information that these tumors have the activation of these pseudoepoxic pathways. Then based on the molecular phenotype of these tumors, we also decided to test a combination of two drugs that had never been tested against these tumors. And we did some studies in vitro on cell lines and in vitro also on primary cells from our rats or from patients. This was a combination of PI3K inhibitor and a CDK4/6 inhibitor. And then we did some in vivo studies and everything actually pointed out to the combination as being highly effective at reducing tumor growth in vitro and in vivo. And then the next step would be to now treat with this combination our MNX rats in vivo. So we used at the beginning of this SFB, when we were again characterizing these tumors, we used several imaging modalities to make sure that the tumors could be visualized with the tracers for PET imaging that are currently used in patients, in pheochromocytoma patients. So we tested various tracers, and we could visualize the tumors with all these tracers. And this, in a way, was making us more confident that we could that these tumors actually could be used to model the human tumors. And I would like to just briefly talk about one of the tracers that we evaluated. So several years ago, Professor Schweiger actually suggested that we test this LMI 1195 in our animal model of pheochromocytoma. This is a MIBG analog, so it's a norepinephrine analog. And so it was developed for cardiac imaging, if I remember correctly, and had never been tested in these tumors before. And so, because of having the availability of this animal model, we tested our rats, and we could visualize the adrenal tumors of the rats very well. And this study, as I said, was published already a few years ago. And so these tracers seem to be very specific for these chromaffin-derived tumors. And I mentioned this because I was very happy to see that actually this tracer was taken by colleagues of the nuclear medicine one step further, and they actually evaluated this in vivo in patients with suspected pheochromocytoma and paraganglioma. And and the conclusion was actually that this LMI PET could be effective as a staging tool for suspected pheochromocytoma and paraganglioma. So this, for us, let's say more translational scientists, was a nice example of a contribution that we could give with our animal model in terms of identifying a PET tracer that might be suitable for these patients. Then another tumor type that we work with is pituitary tumors. They also develop in these rats with complete penetrance. And Specifically, these tumors belong to these non-functioning pituitary tumors, so they don't secrete hormones. And so in patients, they get diagnosed relatively late when they are already invasive, and so the complete surgical resection is not possible, and they tend to recur. And they don't respond to standard therapies with somatostatin analogs that usually target, like octreotide and lanreotide. They are the gold standard for functioning pituitary tumors, but for the non-functioning, they don't work so well. And these somatostatin analogs, as you well know, are mainly targeting somatostatin receptor 2. So for these tumors, there is a need for novel therapeutic targets or novel therapies. And so we recently completed, actually this week, we were able to publish this study that I want to present to you, and was a head-to-head comparison of octreotide and pasireotide for efficacy against these tumors. So this is what I told you earlier, that these non-functioning pituitary adenomas, they don't respond to somatostatin receptor 2 directed analogs. And a few years ago, we were able to show that actually, if you look for the somatostatin receptor expression in these tumors in patients, they have a much higher expression of somatostatin receptor 3 than 2. And so this could be one of the reasons why these patients, these tumors, do not respond well to the classical somatostatin analogs. And this actually set the rationale for the study that I will tell you about. So where we decided to test multi-receptor ligand pasireotide that has affinity for other receptors in addition to this. So it has actually more affinity for the receptor 5, but then has also higher affinity for the somatostatin receptor 3 than octreotide and lanreotide. So these are the two drugs that we tested in vivo in parallel in our rats, and this was the treatment plan. So we actually treated the animals with these long-acting release forms for 2 months, and we monitor longitudinally with MRI the tumor growth. And then at the end, we collected the tissues, and we had we did some ex vivo analysis of these readouts. So proliferation, apoptosis, and of course we checked it for somatostatin receptor expression. So as you can see here, basically, if we compare the untreated animals with the octreotide treated or pasireotide treated, the treatments, of course, reduced quite significantly tumor growth in vivo, and pasireotide was more effective than octreotide. And we noticed early on that there was a gender effect in drug response. So from now on, I will present the data from males and females separately. So the drugs were slightly more effective in female rats than in male rats. So here is again the relative change in tumor volume at the different time points when we did MRI for the various treatment groups. And then we saw that the proliferation rates of the tumors actually correlated with the response in terms of tumor growth by MRI in vivo, meaning that the pasireotide was able to reduce proliferation more efficiently than octreotide. And so in terms of the somatostatin receptor expression, of course, we're targeting somatostatin receptors, so this was a key experiment to do. And as you can see here, we were surprised to see that the females at baseline, they have a much higher level of expression at the mRNA level of somatostatin receptor 3 compared to the males. And then upon treatment, actually, we don't see much of an effect except for an increase in somatostatin receptor 1 expression at the mRNA level after pasireotide. The rest basically pretty much stays the same, so not these two compounds, they don't affect the expression of all these genes. Then we also did, of course, some immunohistochemistry to validate, let's say, to look at the protein level, so the expression of the different receptors, the ones that at least can be detected reliably in rat tissues. And basically we saw a decrease, although this didn't happen at the mRNA level, but at the protein level there was a decrease in somatostatin receptor 2 upon treatment with these 2 compounds. And this actually is in line with what has been reported in patients. But the bottom line is that this, what I showed you earlier, so that these difference in the somatostatin receptor 3 expression, which also was confirmed at the immunohistochemistry level, basically can be the reason why there is a difference between the 2 genders in the response to these compounds. And we also saw that usually somatostatin receptor 3 has been associated with, when it's activated, to the induction of apoptosis. And we saw shrinkage of some of the tumors in the females, especially after pasireotide treatment. And we could show that this is associated with an increase in apoptosis. So because of this potential link between a somatostatin receptor 3 expression and gender, then we looked also in a previously characterized and previously published cohort of patients, of non-functioning pituitary tumor patients. We had never stratified for gender at the time, and we saw that in terms of somatostatin receptor 3 expression, that the female patients, they tend to have higher level of somatostatin receptor 3. This was done by immunohistochemistry than the males. This is just a trend, is not significant, but this could be potentially useful for patient stratification in view of a therapy with pasireotide. So I want to finish with just one study that also we did within this SFB in collaboration with several other people. And this led to the identification of a novel therapeutic target for these pituitary tumors. And it started again from our molecular studies. We did transcriptome of these rat data. We identified some markers that we validated in the human tumors. And then we went back to these data and tried to find additional interesting potential therapeutic targets. And so, as you know, one of the important processes, one of the hallmarks of cancer is tumor angiogenesis, and then, the best known player is VEGF, which is secreted by a lot of tumor cells, and this supports neuroangiogenesis. But there are other molecules that are actively involved in this process, and they are angiopoietins and the Tie2 receptor. This, let's say, this pathway is also actively promoting neuroangiogenesis and endothelial cell survival, and it's been really well characterized in endothelial cells. So these cells are able to secrete these cytokines, these angiopoietins, angiopoietin 1 and 2, and then bind to the Tie2 receptor, and this promotes survival of endothelial cells. In tumor cells, was not really, this has not been really looked at very much. So based on our molecular studies, we found that the rat pituitary tumors, they have a high expression of angiopoietin 2, which is when it's present at high levels and higher than angiopoietin 1, then together with VEGF can promote angiogenesis. So the signature was suggestive of increased angiogenesis in these tumors, and this was validated at the mRNA level and the protein level. So this was the situation. I mean, this, I'm summarizing a few years of work, but so the high expression of angiopoietin 2 we found in these pituitary tumor cells. Then we were able to demonstrate that these cells also expressed the Tie2 receptor, which was actually unexpected. It had never been reported to be expressed in other cells besides the endothelial cells. And so the question was, okay, so these cells can also secrete angiopoietin-2, and yes, they can. And does it work also as an autocrine stimulatory factor as it does in endothelial cells? And again, we could show all of this, and we could show that this angiopoietin-2 type 2 signaling actually supports cell viability in vitro and in vivo. And we used then drugs to block this pathway in vitro and in vivo. We did actually 3 different in vivo therapy studies that were very important to convince the reviewers that actually this pathway is a interesting potential therapeutic target for these tumors. And basically these studies found this new pathway active in the tumor cells, and because of the expression of the type 2 receptor, basically these cells can then respond to angiopoietins that are in the microenvironment. So this pathway is involved in the crosstalk between tumor and endothelial cells. Is a target for therapy, so that if we block it, then we saw a decreased proliferation and survival of the tumor cells in vitro and in vivo. And with this, I stop my presentation and I come to the acknowledgements. And I think that my big thank goes really to Professor Schweiger for inviting me many years ago to be part of this family, actually. And over these 3 funding periods, I've learned a lot. I've established a lot of collaborations, and our projects really took advantage of this great network of expertise that, of course, is not our primary expertise, and also all the infrastructures that were provided by the SFB, and this led us to all these in vivo studies that otherwise we would not have been able to perform. And so I want to thank all the people who are here, who were involved in the studies, and all the ones who were previously part of the SFB and already left and moved on. And of course, the people from my lab for doing the actual work. So thank you for your attention.
Thank you very much for your interesting data. I have a short remark. There is at least one very good SSTR3 selective somatostatin antagonist.
Yes.
And I would foresee an interesting study if you can upregulate the receptor even with pasireotide and then follow up with targeted radiotherapy. That could be one of the future projects.
Yes, so we actually, I didn't have, for sake of time, I didn't include this part, but we have also in parallel evaluated a novel somatostatin receptor 3 analog that was developed by Italfarmaco in Italy. And this actually showed an incredible gender difference, so that the females really responded as when we were expecting based on the higher level of somatostatin receptor 3, and the males, basically, there was no effect. So this data is also being summarized now for publication. Thank you.
Thank you. More questions? If this is not the case, I guess we have a change in program.
Yeah, we have decided, because time has advanced, that we break now for the lunch, because there's a fixed time in the afternoon when Ralf Weisleder has kept his schedule open to give a presentation here, so we have to rearrange a little bit. So the lunch will be served outside, and due to the corona situation, this is not a free buffet. You get little lunch boxes. This is the only thing we are allowed to distribute here. So please enjoy the lunch boxes. And I like to mention that the boxes are prepared by a company across the street. It's called Tellerrand. This is a company he is supporting active refugees in Munich, and they are actually participating in this lunch service, and we use it as often we can to support this activity across the street. So only as background information, please enjoy.
So we will continue with the next talk by Johannes Notni, and he will tell us how to make all these tracers Bernd Pichler is asking for. So please, floor is yours.
Yeah, thank you for this really, really encouraging introduction of my person. Well, I'm not able, of course, to make all the tracers, just a few of them. And I noticed that in Bernd Pichler's talk there were a lot of interesting new targets mentioned, but actually integrins was none of them. There was just a tiny, I have seen it, there was alpha V beta 3 integrin mentioned somewhere in really tiny letters, but actually, yeah, but actually there are more integrins than that. You see, there are the 24 integrins, and here is a slide which shows you for which of these integrins there were actually tracers being made. This only concerns radio tracers, of course, and which of them already went to clinics. And you see, it's just a few of them. And I think an estimate of 99% of these tracers were dealing with the alpha V beta 3 integrin. But this, of course, this is the integrin project. I call it the integrin project because since I went to TUM in 2009, I was more or less constantly working on this integrin. And in this moment, I have to really thank Markus Schweiger because in 2016, he asked me to set this up as a whole new project in the SFB for me as a PI, and so I could continue this on a regular basis. And I don't want to dive too much into chemistry, but when it comes to integrins, of course, a big benefit we had here at TUM was the group of Horst Kessler. And basically, most of these ligands have been made by the Kessler group. There's one exception. This one has been done by the Marinelli group in Napoli. And I had the luck to have access to all these ligands and make radar tracers out of them, which I'm really grateful to Horst Kessler that he gave me this opportunity. And on the other hand, when it comes to radiolabeling, here's a molecule which keeps me busy for now, let's say, almost 15 years. 2008, I think was been first made, and I'm also quite happy that Helmut Mäcke is the very person who chaired this session, because I remember quite exactly when I was presenting first time this molecule on a conference in Warsaw in 2009. I was really enthusiastic, and he told me, oh, well, this is a nice ligand, but DOTA and NOTA will do well. And after all this time, I have to admit he was probably right. DOTA and NOTA is really fine. So it was not so exceptional as I thought back then. But there's something to it which makes it unique for this particular application. And this is you can make trimers out of it. You see, it has 3 sidearms. You can make trimers quite easily with different synthetic routes. And this was what's driving the project, because this was, here I presented the outset of this integrin project before we even started to do this in the SFB, at least as project A10. And you can see what this trimerization of these ligands can do. Here's a monomer, here's a trimer. This is for alpha V beta 3, this is for alpha 5 beta 1 integrin. And you see, we get much better tumor background contrast for at least this situation here, and we get contrast to the monomer, to the trimer. It gives a much better tumor retention in both cases. And this encouraged us to go further with this concept. Just to mention, for all these tracers, we could imagine a lot of different applications. One idea we're just pursuing at this moment is that we could probably use it for imaging angiosarcoma. This is an idea which came up from the pathology department, of course, which is hosting me since the beginning of 2019. Here's another part of this story, alpha V beta 8 integrin. You can see the trimer. Again, you get in the same animal much better tumor to background contrast. Here the retention is not improved as good as for the other examples, but nevertheless, it worked out. And alpha V beta 8 integrin was just a side story for this project, because there's not really much data what alpha V beta 8 integrin imaging could be good for, after all. We did first in human translation and saw a quite unique distribution pattern with uptake in the neuronal tissue up here and there. But we still don't know how to interpret this, and I think this is something we will probably do in the future. But now, let's switch to the really important story, at least important for this A10 project, which is alpha V beta 6 in the Grin. And alpha V beta 6 in the Grin is interesting in that it is expressed on epithelial cells and absent in adult tissues, at least according to immunohistochemistry data. And the interesting part of it is that it is, for instance, expressed to a certain extent on the cell surface of pancreatic carcinoma cells. And it is said that it's associated with the invasive growth of these tumors. You can see that here. You see particularly dense expression on the basal membrane in these pancreatic carcinomas. And of course, I have just 20 minutes and should keep it short. I was just being told, so I will hurry up. This is essentially the whole development of this tracer we did in A10 over the last few years. We started out with the tracer, a monomer of this alpha V beta 6 integrin binding peptide, and you can see there's just a faint shade where the tumor is. If we trimerize it, we get a good signal for the tumor, but of course, this is quite obvious that it's not a good tracer. And we ended up with a lot of liver and lung uptake here, although tumor retention was fine, but blood clearance was really slow. And this is what happened during A10. Essentially, we got rid of all the unwanted uptake except the kidneys, which is to a certain extent excretion related. And we get nice blood clearance, nice tumor. And now you probably might want to ask, how did we do that? And this is something my PhD student Neil Quigley, he's over there, he did in the last few years. And I try to summarize his results in the most appealing manner. So this is this trimer we started out with. And you can see here, I just put out for you the important part of the biodistribution. This is liver, and this is the control, and just right-hand side is the blockade. You see we have a certain blockable uptake in the tumor to a certain extent, and all the liver uptake we see for this trimer is fully blockable. Now, what we did first was to exchange one phenylalanine versus a tyrosine on that construct. Looks like this. And essentially, exchanging a phe by a tyrosine means just to add a hydroxyl group over there. This is just a tiny chemical change. And what you can see here, you see the symbols down here will show you, guide you through to hold this process. What you can see here, the liver uptake steeply decreased, tumor uptake stayed more or less the same. Now there are different options to, of course, to exchange a phe on a tyrosine on this whole trimer. On the other side, on the right-hand side, you see this effect was even more pronounced. And at that moment, I'd like to want to step back just for a second and realize what is going on here. We have a molecule of 4.3 kilodalton, and we insert into one existing CH bond over here, we insert an oxygen. This is just 16 daltons. We increase the mass of the molecule by less than 0.3%. We don't change the log D, and still half of the liver uptake is gone. That's quite a surprise. So what happens if we exchange both of them? Funny enough, liver uptake stays the same, tumor uptake stays the same. Of course, there are different possibilities to put two tyrosines in this molecule. This is one of them. You can see here. Again, liver uptake goes down. And this, you see, this was the side with where the effect was less pronounced than on this side. If we do it on the right side, you see the same pattern here. This is exchanging one of the tyrosines, bad side, good side. Here is bad side, good side again for two of them. And still, there's no effect in the log D, which is normally predictive for something like liver uptake. So, quite interesting. I guess you all follow this situation over here. The blood clearance gets better and better. Again, if we do this on 3 different phenylalanines exchanged by tyrosines, you can see liver uptake is even further down. And again, on the other side, the effect is more pronounced. But still, now we see effect in the tumor uptake, and we see an effect on the log D. When we do this with 4 tyrosines, you see we probably have reached a bad end here. Maybe liver uptake is further going down, but still, as it seems, tumor uptake is well, going so much down that it's not worth going further in this direction. But again, a surprise, if you exchange all of those phenylalanes with tyrosines, you see tumor uptake is up again, liver uptake reaches a global minimum, and the log D is somewhat going down, so it's more polar than, but still we end up with a quite appealing biodistribution. And I think what we learn from this situation is that basically none of these effects could be foreseen from the properties of one single of those peptides. You see, this is all multimers, homotrimers, heterotrimers, but what they do, if you combine them in this different manner, you cannot determine from the components. So we have certainly some emergent phenomenon here when it comes to relate the structure to the biodistribution. So to cut this long story short, this is what we ended up with. So the final compound with the 6 tyrosines we call trihexene. And I wrote here down the highest tumor and best PET contrast of all alpha V beta 6 integrin PET traces. So how can I claim this? And simply we compared to the recent literature. This is the literature I know where such traces by other groups went to patients. It's all of 5 different traces. And you see, we took the best one of those and compared it to the preclinical data for this tracer. And I think you're still tired from the lunch, and so probably you won't have a look at those numbers. Just look over here. You see, without tracer, we have a much higher tumor uptake than the best in class of the competitors, and also a much better tumor to pancreas ratio. And I also like to mention that this is not because we used a really highly expressing tumor model. Actually, if you have a look at the immunohistochemistry down here, this is quite close to what we know from the patient situations or from the PDAX samples I showed some slides before. Now again, seeing is believing. This is some images I took from the literature, and you see this is probably something which was not really encouraging to translate these traces or really go for a broader clinical use, because you can see they have all this intestinal uptake, and in the first human image we did with the trihexene, we found all this uptake is essentially gone. This was quite encouraging, so we see that we don't want to see, we don't see what we don't want to see. And the question is, do we see what we want to see? And this is, of course, something which you can nicely see here. With the trihexene, for instance, we can really nicely detect a head and neck tumor. And over here, you see in the FDG scan, we have a lot of lymphatic uptake, and probably this is maybe related to inflammation, all of which we don't see here. Of course, this is just anecdotal evidence at the moment. We cannot tell whether this is really more specific, but at least these images made us believe at this moment that it could be the case. I was talking about pancreatic carcinoma, and here's an image of a patient with an isolated primary in the pancreatic head, and you can see there's a really nice contrast to it, but of course detecting a primary of pancreatic carcinoma is probably not as important as detecting metastases. And this is what we also could do. We have here metastatic pancreatic carcinoma and could really nicely detect some metastases in the liver. And at this moment, of course, you see this was 120 minutes, and with 100 megabecs, I really wished we had put this patient into this total body scanner. Probably then we would have got a really state-of-the-art image. So I'm really looking forward to have one of these scanners available at one time. And here's another head and neck tumor. It's not a squamous cell carcinoma. It's a recurrent metastasis 2 years after surgical resection of the primary. And this case is interesting because this metastasis has been dissected, and we could actually analyze it via immunohistochemistry. And we saw this pattern we are also already familiar with, and which is quite consistent with the theory that in the central area of the tumor we have a little bit less expression than in the infiltrating area. So apparently this integrin is really showing us some of this infiltrating infiltration behavior, this invasive behavior, so it relates also to the biochemistry of the tumor. And instead of a conclusion, I will just present you this comparison. Well, I think it's coming back to the start of my talk. I think it's important to note that probably integrins as emerging targets are really undervalued. I think there's much more to integrins than it has been believed during the last 20 years. And to sum this up, what my experiences here, since I came here and made the first integrin tracer for alpha V beta 3 integrin, and here's one for alpha V beta 6 integrin, you see they have been all in the slides, structurally really similar. This is probably what you wanted to see all the time for alpha V beta 3 integrin, but alpha V beta 3 integrin has the problem that there's all this physiological expression here, all this off-target signal, and this, of course, cannot be avoided because it's not due to the tracer, it's due to the physical target expression. And this is probably what we all wanted to see all the time. So I think we should give it a second try with the integrins. There should be more to it. Yeah, I just said that I'm here for 12 years now and done all the time together with a lot of people, all this research, and it's impossible to remember who has helped to pursue this project all the time. So I'm bound to say that I have to thank all these people from the institutions and all the collaborators, because at some point, I think everyone has done us a favor in one or other way. And I thank you for your patience. Listen to me.
Thank you very much. Very, very impressive. I have a short question. The chelator, how good is it for lutetium, for instance? Can you label?
It does not work at all.
Does not work at all.
There's no way to put lutetium in, but we have this larger congener, this DOTA-like analog, we call it .P, and this works for lutetium.
That works. Okay.
And needless to say that we're doing this right now.
Okay, good. Questions from the audience? Please.
Short question to the synthesis of the tyrosines. So are you starting the synthesis anew to replace the tyrosines? Or can you modify the phenylalanines after you synthesize the larger molecule?
It takes me a second of clicking while I'm talking. Well, actually, it's way simple. Because what we do is we can click these different peptides on the chelator. And for instance, if you go simply for stoichiometry, you take 2 peptides on one chelator and will give you the 2-by-1 result. Of course, it will give you a mixture of the others as well. And then you put there another peptide, which you have synthesized before, and you end up with a mixed multimer. So, I mean, this looks like a whole unit. And it is, biochemically, is a whole unit. But chemically, it's just a trimer where you put the individual peptides as you choose. It's quite straightforward.
What made you to go from phenylalanine to tyrosine? What was the original idea?
Basically this. We thought, okay, this one is not good because it's probably too polar. Too non-polar, and we said, okay, so by any means necessary, we need to add some polarity to this molecule. And then, when this peptide, the original one, where is it? Where's one of them? When this was described, this was the headliner of some publication, of the first publication in 2016 from the Kessler group. And they presented this as the most selective variant for this alpha V beta 6 integrin. And actually, they had also the other peptides with monotyrosines. They also had in this paper, but they didn't show up on the front page. They were in this supporting information, but they were just slightly less effective and slightly less selective. So it was somehow, well, obvious to do it. What they didn't use was, where is it? That peptide. This is something we simply synthesized and found out it's not much worse. And as a trimer, it works perfectly.
Okay, if there are no more questions, thank you very much. And let's come to the next talk, which is by Professor Skerra from the project A8. Please.
Yes, thank you very much. And in particular, thank you very much, Dr. Schweiger, for inviting me many years ago to join this SFB, and also for having this last phase here as co-PIs of Project A8, which was very constructive. And actually, I will have this as a joint presentation together with Volker Morath, who is actually the project leader in this project. Now we have a totally different subject here because we are not medical doctors or tumor biologists or medicinal chemists or radiochemists, we are protein designers. And in fact, my lab has made a couple of contributions to the area of protein design over the last 30 years. Some of our technologies, like the anti-kaolin technology or puzzylation technology, actually even inspired some spin-off companies. And at the moment, 5 of our designed proteins are in clinical studies up to phase 2, and more are to come. Now, what the question was actually when Professor Schweiger asked me to join this initiative, I must say I didn't know much about nuclear medicine and in vivo imaging at the time, but together we had the feeling that some developments from my lab might contribute to provide better reagents in terms of protein tracers. Because at the time, I mean, we had essentially the existing technology 10 years ago or so was just having classical radioimmunotherapy or diagnostics based on antibodies that were more or less randomly labeled with radionuclides. And this is something which for a protein designer is not ideal. I mean, first of all, as all of you know, and as already has been mentioned this morning, antibodies are large molecules, they have a very long circulation. Also, they are difficult to handle under GMP conditions. They are so-called complex biomolecules. And also the fact that in many cases radionuclides are more or less statistically conjugated to the antibody also creates an additional complexity, both in terms of manufacturing, in terms of quality control, but as we also have learned in the area, in the closely related area of antibody drug conjugates, by now it's well established that the place, the site where the, let's say, toxic or here radio component is linked to the antibody can make a huge difference in terms of specificity in vivo and also activity, depending on the application. And this is totally different with our designer proteins, which are smaller, smarter, easier to manipulate, easier to produce. And just as one example here, this is one of our typical mass spectra. So we have molecules that are precisely defined on the atom. It's more almost like with a small molecule. Whereas with these classical molecules, you always have mixtures, okay? Here we have very well-defined molecules. And as these molecules are very small, so we talk here about antibody fragments or alternative binding proteins, anti-kaolins as we call them, or flexible linkers, we can actually combine them. We can manipulate the size of the molecule, and by this means we can manipulate the circulation time. We can introduce single reactive groups for site-specific labeling, radiolabeling. We can combine different binding modules to make bispecific molecules or trispecific molecules. I mean, of course, these buzzwords are not so unique. You see them in many other publications, but I can tell you, in most cases, this is only paperwork. In our cases, these molecules are truly well-defined and nicely behaved. And I will just give you a glimpse of the 3 main protein technologies which we introduced here, and then Volker Morath will talk a bit about some applications which we were able to realize over the last years. Now, the first technology is the bacterial production of recombinant antibody fragments, which I myself pioneered 30 years ago, and which is very useful to make proteins quickly rather than producing them in cell culture, and helps us a lot. And proteins made in our lab fermenter actually have already found application in so-called Heilversuch. The other technology, and which is actually true protein design, is the creation of a new class of binding proteins, which we call anticalins. And you see them here drawn to scale in comparison with a full-size antibody. And as you can see, these small proteins have a binding site for an antigen, which is very similar to the binding site of an antibody, but they lack all effector parts. They are made only of a single polypeptide chain, so you don't have to deal with two different genes or link them somehow. So these are very small and robust proteins. In a certain sense, comparable to nanobodies, with two important differences. First, we don't have to immunize camels because we make them from synthetic libraries, but the scaffold, that means 90% of the protein, is based on endogenous human circulating proteins, the so-called lipocalins. And as clinical studies of our spin-off company peers have already shown, under therapeutic settings, we have extremely low immunogenicity with our proteins. And this is just a collection based on X-ray crystallography, so molecular imaging, so to say, where we can show that we can generate binding proteins against various targets, from large proteins over peptides, including Alzheimer amyloid beta peptide, and also to radionuclides. So this is an yttrium DTPA complex where we have a very tight binding, and tight means picomolar dissociation constant, very tight binding anticalin. Volker Morath will give an example. The other technology which we have introduced here with some benefits is PASylation. PASylation is a biological alternative to pegylation. You are probably aware that pegylation of biopharmaceuticals is a currently approved way to prolong circulation half-life. And in fact, we also had the feeling that somehow tuning the plasma half-life of small proteins could help in vivo imaging, but we wanted to get away from PEG because PEG has a lot of disadvantages. And I can tell you, after the current COVID vaccination campaign, as you know, all the mRNA vaccines contain PEG. I believe that the future of pegylated biopharmaceuticals will be short because many patients will develop hypersensitivities against PEG. So our technology is not based on synthetic polymers, but just on 3 amino acids, proline, alanine, serine. And this makes long polypeptide chains with biophysical properties extremely similar to PEG. As you can see here in this computer animation, here we have an FAB fragment with its binding site. Here we have such a long pass chain. This can be 200 residues or up to 800 residues. And due to Brownian motion, these naturally unfolded and structurally disordered polypeptide chains wave around, and on time average, it looks like a cloud. So this makes proteins appear bigger than they actually are, and this effectively reduces kidney filtration by up to 2 orders of magnitude. And the first proof of concept that this can actually help for in vivo imaging was achieved by Claudia Mentler and also with the help of Professor Wester, where we made PASylated versions of an FAB fragment directed against the well-known HER2 antigen. And it's very simple with our gene cassettes to make fusion proteins with pass sequences of different lengths. So the longer the pass sequence, the larger is the protein size, and the longer is the circulation time. And you can see here that the tumor accumulation of the iodinated naked FAB fragment is there, but the picture is somewhat blurred. But it becomes much better, more intense, and with better background ratio with increasing pass chain. And then, if the pass chain becomes too long, then the overall behavior of the FAB more or less looks like an antibody in circulation. So at least in this time window here, 24 hours post-injection, the protein was not yet eliminated sufficiently from circulation. Yes, and based on these technologies, which I briefly introduced, we provided here a couple of binding proteins against various tumor targets, either anticalins like against PSMA or CD98HC, or Fab fragments or single-chain FE fragments, and so on. And Volker Morath will now show you a few examples how this looks under in vivo imaging conditions.
Thank you a lot. So it's a pleasure to now go to the results that we obtained in this funding period. And at first, I would like to give you a short outline what I will present to you today. And I have grouped the results in 5 different categories. At first, we were working on novel binding specificities of anticalins that we transferred to preclinical research. Then we also had a more technical part where we optimized half-life by pacylation and worked on bioconjugation and novel radiolabeling techniques. Then we also tried to do clinical translation in so-called Heilversuch experiments. And then for the end, I would like to give you some insights into the latest results from our pre-targeting approach and a novel project that we started. So at first, this is an anticalin that was selected in this funding period by Christian Deutschle, and it's targeting CD98 heavy chain. So this is a part of the heterodimeric amino acid transporters that are expressed and overexpressed in different cancer types. And the interesting thing here is that this high affine anticalin was selected directly from a naive library by phage display, and down there you see the crystal structure that was solved by the lab of Professor Skerra, and this here is the target that is tightly bound by the anticalin. Then from Weinstefan, the new specificity was moved to Munich, and we then received a radioligand that had the increased half-life by pacylation and was conjugated with DFO and zirconium labeled. And then we performed preclinical studies with PC3 xenograft tumors, and we did imaging at 24, 48, and 72 hours, and could see that the tumor can nicely be visualized, and best contrast is achieved at 24 hours, and we can also block this signal, as we can see here, compared to the unblocked samples. Then, what Professor Schweiger mentioned with these smart targets, 2 years ago, before this whole COVID thing, we were here and listening to a presentation of Florian Bassermann, and he found out that IMiD therapy somehow is having an effect on CD98 heavy chain and other membrane proteins. And during this project, he found out that this is due to interaction with chaperones, and he was interested in doing imaging with this anticalin that binds this CD98 heavy chain. And so we used this anticalin also to analyze treatment studies of patient samples, and also did again some xenograft studies with multiple myeloma. Then, besides CD98 heavy chain, we also focused on PSMA binding anticalins that were developed in the last funding period. That in this funding period, we then wanted to compare different formats in which we matched the physical decay half-life of the radioisotope with the plasma half-life of the protein. And so we came up with 2 different formats, one being the anticalin just labeled via cysteine residue with no DAGA, and on the other hand, plasma half-life, the extended version, where we used the linear chelator for zirconium DFO. And you see after 1 hour, we see the tumor, the LN cup tumor, imaged by the anticalin with the gallium variant of the radioligand. And when we then compare this to the zirconium and pacylated radioligand, we see that the accumulation in tumor is double as high when compared to the gallium variant. And again, the time point at 24 hours seems to give the best results. In this experiment, we also wanted to compare 2 different chelators. So one, the DFO, which is at the moment the most used chelator, with fusirenin C, which is basically the same but just circular, and this was provided by Clemens de Cristoforo. And we did a direct comparison and found out that the demethylation in vivo is significantly decreased when using this cyclic radiochelator. So this is something that could be used in the future. Then the third point I wanted to point out, because there is no presentation by the set projects, I shortly wanted to introduce the effort of trying to do Heilversuch. So we wanted to transfer proteins that were developed from the SFB into the clinic and do initial imaging. And the first Heilversuch started directly at the beginning of this funding period. And there we based a radioligand on a trastuzumab-derived Fab fragment for HER2. And this was then tested in a single patient. And we could see the primary tumor as well as some lymph nodes. And currently we are working in collaboration with Calogero and Alexandria on a next Heilversuch where we want to image a Fab fragment for GAL3 for thyroid cancer. And this is some instrumentation that we got for the cold room so that we can now purify these proteins in the cold room. Then the last topic that we were supposed to do in the SFB project is pre-targeted radiotherapy. So this is now a combination, and we want to take for therapy the best out of both worlds. So we want to take the advantages of small molecules and proteins and combine them to have a better therapy at less side effects. And this works by at first administering a protein that is bispecific. One of them is binding to a tumor antigen, and it's just a protein that needs a long time to be eliminated. In this time, a lot will accumulate in liver and in kidneys. And when then this protein has cleared from the circulation, then we apply the radioligand. And the advantage of the radioligand then is that it's rapidly excreted from the circulation and gives less dose to the off-target organs. And this was the starting point, so to say. So these proteins are binding to CEA, the tumor marker, and then we have this mentioned before anticalin that binds to the radiochelator DTPA. And now these are some first recent results. We produced again the protein and then used now the small animal SPECT CT scanner from Mediso that is in the basement of the TranslaTUM. And we applied or administered the protein, and afterwards we had a dimer that we injected together with lutetium, and we could see nice accumulation in the xenograph tumor. These are LS174T tumors that were above the shoulder, and we see a nice accumulation, and there's no signal to the kidneys as we would expect it for direct labeling, and we only have a minor problem of some radioligand that goes to the colon. On the other hand, if we don't add any protein, so no protein control, doesn't give any targeting of the dimers to the tumor, and this is also to a certain degree stable over time. So at 6 hours, there is pretty much no change in the whole uptake. And having said this, and this was quite encouraging for us, this is still somewhat non-ideal, because the anticalin also has a preference for certain radioisotopes, and the diameter of the radioisotope has a huge influence on the affinity, and here lutetium is by far not ideal. And also, this is just monovalent interaction, as we saw before in the talk from Johannes. So to move this pre-targeting approach, and I guess I could convince you that it's working with this proof of principle in vivo experiment to success, SPECT, as I showed you, is an important point, but also protein engineering, so to have polyvalent binding proteins that we've already produced is a key, and as we heard before in the talk of Johannes, also optimization of the hydrophilicity of the ligand. And then what I found out recently, that also nuclear medicine is somehow part of this PRISMAP, and so we can now get also some innovative radioisotopes that are quite ideal for this anticalin. So, and this is something that we just developed in the last years because we were interested in reporter gene imaging. And here the principle is basically the same. We take the anticalin that binds the DTPA, but we don't produce it in E. coli and apply it as a recombinant protein to the patient, but we rather put the gene into the patient by CAR T cells or some gene therapy, and then the host cells will produce the protein, and then we came up with a radioligand that can then be labeled with F18 and can detect these reported genes. And these are the results that we obtained. So this is just a dynamic scan of a mouse that had 2 tumors, one with the specificity of DTPA and one with another random anticalin. And in the end, we see nice accumulation of the tumor. The signal stays the same. Everything else, all organs and tissues have low uniform background. We only see minor excretion-related signals. And here we had a look on AAV9 gene therapy. So we produced these viral vectors that encoded for the reporter gene. And this is state-of-the-art, what people cite at the moment, so bioluminescence imaging. And we could then see all the transduction that happened in the mouse. So the myocardium, which gets specifically targeted by the virus, arterioles, and the kidneys. And this can then also, in collaboration with Katja Steiger, be imaged on a cellular basis. So, and I hope I stayed in time. I would like to thank all the advisors and the mentors of the project, especially Markus Schweiger, Arne Skerra, and also Wolfgang Weber. Thank you.
Thank you very much. Very interesting data again. Questions from the floor? Short question. Your pre-targeting approach, does it, you indicated something like terbium, the terbium DTPA compound may bind better, has a higher affinity to your
yes, I mean the isotopes that fit perfectly are yttrium, gadolinium, or terbium.
Okay.
And yttrium was not suitable for SPECT imaging, and terbium was basically not available, and gadolinium there's no radio.
But the metal ion does not play a role, right, on the affinity.
The anticalin binds the complex, the DTP.
Yeah, I understand.
Yes, and the radius of the central ion plays a role
It does.
in the overall size of this complex.
Okay.
And this influences the affinity.
Okay, good. More questions, please.
You showed that the FAB fragment labeled with iodine, the naked one and your complex, were quite similar in the mouse picture. Your complex does prevent from dehalogenation, or is it the same problem?
No, the effect of pyrrolation is more or less only the circulation. So if you give, if the protein has more time, more contact time with the tumor, that's our hypothesis. If it has more contact time, it will also better accumulate in the tumor. And maybe on top of this simple, let's say, plasma half-life effect, there's probably an effect, as you know, enhanced permeability and retention effect. So pyrrolation may even drive tumor uptake. We have seen similar effects in other tumor models, so there may be a general phenomenon. However, if you make the size of the protein too big, then one has the problem with background from vascularization.
Thank you. If there are no more questions, we conclude the session 2. Professor Ulrich Keller is giving the final talk on
Hello, good afternoon. It's a really great pleasure to be here. Professor Schweiger, thank you very much for the invitation and this fantastic final symposium within the SFB. So my presentation is entitled Targeting Mäcke, Associated Lymphoma Biology with Probes, Drugs and Cells, and I chose this title because of the fact that I moved away and did not have all the fantastic possibilities and opportunities within the imaging consortium any longer. So you will see we hopefully did other very interesting things with the funding we received. So what is MYC? Most of you, I guess, know it's a transcription factor. It's a very frequently aberrantly expressed oncoprotein. Usually at physiological levels, it targets so-called EBOX target genes, but when expressed at high levels, as we find it in cancer, it basically targets also non-EBOX target genes, which can make up to 15 to 20% of the genome in the end. What we know is that MYC is a central driver associated with aggressive cancer biology, poor prognosis, and treatment resistance. This is the pan-cancer view. When you just take a look at CBioPortal, at all cancers, you find that about 5% of cancers show copy number alterations of MYC. And in basically every single entity, these alterations are associated with inferior prognosis. You also can derive from these data in CBioPortal something about the biology MYC is associated with. For example, you find that it's frequently associated with loss of p53, and this is pretty obvious. MYC drives proliferation, induces apoptosis. When you lose the p53 apoptotic axis, this of course collaborates in cancer pathogenesis. This is the look at lymphoma now, and you can see that up to 10% of human lymphomas show MYC genetic alterations. And again, and it's a little more complex than shown here, this is associated with inferior prognosis. So just a very brief overview. What does MYC cause in cancer? Well, it affects the cell cycle. It causes immune escape, a field we are also working in now. It affects the tumor environment. It is associated with causing genetic instability. It induces apoptosis and collaborates with defective apoptosis. And something I will now go a little more into detail, it also is, of course, involved in growth and metabolism. This is the workhorse model we are using in my lab for several years now. It's the emu-MYC mouse, where MYC is expressed under a promoter that causes a massive overexpression of MYC in B cells, 50 to 100-fold, as shown here in the transcript analysis and protein analysis. And what happens in the early stages of mouse life is that there's a balance between increased proliferation and MYC-induced apoptosis. However, during the course of life, during the first week, 2 months of life, apoptosis is not functional any longer, and there's pathways and mechanisms are induced that are important for cancer pathogenesis. So one very early work within this SFB was together with Andreas Buck, who then left, and Jürgen den Hollander, who is now in Bern, a neuroradiologist, where we found that MYC, in fact, induces mitotic kinases, aurora kinases. And when you target these mitotic kinases by small molecule inhibitors, there's a synthetic lethality between these inhibitors and MYC-induced expression of these kinases. Some more work from the earlier funding within this project is shown here. One very important target of MYC is also DNA metabolism, in particular thymidine kinase. You who are in the imaging field know very well that there's one very interesting agent to image proliferation, and this is FLT, shown on the lower part of this slide here. And what we did here, and this was a collaboration again with, and it's only shown one of the many things we did with this tracer within the consortium, is a work which showed that you can use FLT as a tracer to differentiate between glucose metabolism measured by FDG and proliferation measured by FDG. This is shown here, collaboration with Andreas Buck and Clemens Schmidt in Berlin. It's very unfortunate, I think, that this tracer did not make it further in clinical development. I hope there will be some studies in the future, because FDG PET often does not help us to differentiate between a tumor that is there, that might be senescent, and a tumor that still has the potential to proliferate within the next months or years. So I think there's some possibility that there will be a revival of FLT. So about when the last funding period started, we changed our strategy. Previously, we usually took a biased approach. We knew, based on genomic sequencing data available and annotated clinical data, candidates that might be associated with lymphoma pathogenesis or Mäcke as an oncogene. And then we took this biased approach, as I have shown you, for example, with the Aurora kinases. However, there's, of course, a risk by doing this because you never really know whether the pathway you look at is really important for cancer pathogenesis and thus possibly a biomarker, a predictive biomarker, or a target for therapy. And that's why we changed our approach. We switched to something where you immediately generate functional data. You immediately know from the in vivo model that these data are important for lymphoma pathogenesis in our case, and then you can be pretty sure that you are really looking at clinically or potentially clinically relevant therapeutic targets. This is the model we used. It's a collaboration with Roland Rath, who generated these piggyback transposon and transposase mice, which we used in the studies I will show you now. So that's the cross we did. We started about 4.5 years ago. So shortly before the last funding period, we crossed these 3 mice together. So one lymphoma-prone mouse and then 2 mice which cause hopping of the transposons within the genome of the mice. And as you can see, the first expected result is that by insertion mutagenesis, you really accelerate lymphoma onset. This is the map of the genes that showed insertions. You can see we needed about 50 tumors and found a large number of mutated genes. It's a lymphoma model, and of course we were interested whether these genes are really important for lymphoma. That's why we did an overlay with the cancer gene census catalog, a catalog of genes which contain mutations causally implicated in cancer. You can see there's a substantial overlap, and of course we are interested in novel genes, in genes where the function is not known yet, and genes whose associated pathways have not been implicated in cancer therapy yet. We also did an overlay with then available genes mutated in diffuse large B-cell lymphoma, again showing a nice overlap. So when you do this analysis, of course, you have to validate your findings. Are the genes you find mutated are really important for lymphomagenesis. In collaboration with Roland Rath again, we generated a model where we crossed lymphoma-prone mice with Cas9 mice, did a stem cell transplantation, a viral infection of the stem cell graft, and could then infect stem cells from which lymphomas will then be derived. And in case of a tumor suppressor, of course, we expect an accelerated lymphomagenesis. And I will not show you all the data. This worked. So by doing this and a lot of more mechanistical analysis, we identified several genes and pathways. Just 2 examples shown here. One is D-sumolase, sumo protease, SEMP6, and through a large number of experiments, we identified that loss of SEMP6, which occurs in about 20% of human B-cell lymphomas, results in a synthetic lethal interaction with PARP inhibition. What we also found, and this is not yet submitted, is that another gene, genome maintenance factor, SLF2, which is also affected in about 20% of human B-cell lymphoma, that this might be a biomarker which predicts sensitivity to another small molecule inhibitor here, a sumo inhibitor. So now to the more imaging part of my presentation. This is a bioinformatics analysis of the pathways that were affected in the mouse where we performed the mutagenesis. And you can see here, since we are using a B-cell lymphoma model, of course we expect B-cell receptor signaling affected, but the number 3 and number 1 hit, depending on how you do the analysis, clearly points to chemokine receptor signaling. So these are data from various mouse models that have been published, and we just asked the question which chemokine receptors are mainly affected in these various mouse models, and the chemokine receptor that is mostly affected with or associated with oncogenesis in these lymphoma models is CXCR4. We also did a series of experiments shown on the upper right here, where we showed that MYC directly regulates CXCR4. And in the middle, you can see some more analysis from databases where we clearly find that MYC expression correlates with CXCR4 expression, and that CXCR4 expression also correlates with the translocation of the MYC gene. So what is CXCR4? It's a G-protein coupled receptor. It's important for migration, homing, and in particular hematopoiesis. And the oncologists among you know that we are using an inhibitor of the pathway to mobilize hematopoietic stem cells. This pathway is important for activating a variety of signaling pathways and basically does the biology recruiting cells to a certain microenvironment. It's also a very interesting chemokine receptor because in basically every disease we looked at, whenever CXCR4 is high expressed, this is associated with poor prognosis of patients, as shown here, exemplified in diffuse large B-cell lymphoma. So we had the fantastic opportunity to take advantage of all the platforms and probes developed here in this consortium. And these are the 3 people, Markus Schweiger, Hans-Jürgen Wester, and Margret Schotelius, who have been involved and played major parts in the work I'm showing you now. So what Hans-Jürgen Wester's group did is they started from the structure of CXCR12, the ligand of CXCR4, also known as SDF1 alpha, and developed an imaging probe, which they named Petixa4. And these are some of the images we generated. I think this was patient number 2 or 3, and we first had the chance to image patients before we had the permission to image mice, which is one of the advantages if you do such analysis in Germany. So you can see here that this PET tracer really helps to image lymphoma, and I think it has a huge potential, as shown by a group of the by work of the Würzburg group recently, to become the new imaging tracer in marginal zone lymphoma, clearly outperforming MR and FDG PET, as shown recently. So this is what we generated. Of course, we do not only want to image, but we also want to use something to treat patients, and then took advantage of the meanwhile developed tracer Pentixatea, which can be labeled with yttrium or lutetium to really therapeutically address cancer. These are some of the early studies. Again, some imaging acute myeloid leukemia, a disease where you often have patients who relapse 2-3 times after stem cell transplantation, and you cannot really offer them effective new treatments. What we did then, and these are some of the colleagues who have been involved next to all our imaging colleagues, Stefan Harbringer, Sabine Steidle-Maurer, and Peter Herhaus. What we did, and most of the work was done, and most patients were treated in Würzburg in the department of Andreas Buck and Hermann Einsele, was to really now bring this new therapeutic tracer into the clinic. basically do myeloablation and conditioning of patients before allogeneic stem cell transplantation, which allows you to transplant someone who you would not get into remission with the usually available therapies. You can see these patients, despite destroying their stem cell niche, they all regenerated their bone marrow. And we did, during my time here, not get this into the clinic, but I'm very confident that Peter Heraus and Florian Bassermann will now be able to finally get this phase 1/2 trial, which has funding by Deutsche Krebshilfe, into the clinic. It's basically a theranostic study, imaging the patient and then taking the therapeutic probe to eliminate the cancer before a stem cell transplantation. So I think this study has now a real chance to get started within the next months. What we also did is to investigate some more imaging in patients, and one often diagnostic problem is central nervous system lymphoma in areas where you do not want to perform surgery on or stick a needle into. So what we did here was that we could show that CXCR4 in fact helps to identify lymphoma in patients, and this nicely fitted to a study. We collaborated with the lab of Matthias Heikenwälder, where mainly his group identified the mechanism how CXCL12, CXCR4 interaction recruits lymphoma cells to vessels within the brain. Study performed in the US and in several European countries by a company named Pentixa Pharm, where we now would aim to show that CXCR4 imaging might be a good imaging agent and might outperform other, in particular MR imaging for brain lymphoma. And again, there are plans to also start therapeutic studies. So finally, some final data. I would like to show you the second chemokine we found involved in lymphoma genesis in various models is CXCR5, and not having in Berlin now such a powerful radiochemistry and radiopharmacy available, we will not be able to address this by probes, and fortunately have the possibility and the opportunity now to address this with cells. So we are now investigating CXCR4 in collaboration with Uta Höpken and Armin Rehm, a group at MDC, Max Delbrück Center in Berlin, and they identified that CXCR4 positive lymphoma cells or CXCR4 on lymphoma cells, since it labels only the more mature B cells, but not the cells within the bone marrow, might be an ideal target for an immunological therapy approach. So this is what they did. They generated CAR T cells targeting CXCR5 in lymphoma, showed some nice eradication and efficacy in mice. And this is what we will be starting in 22 now, having received funding from the BMBF for a phase 1 study. We will now bring this into the clinic and target CXCR5 in a phase 1 CAR T cell study, which nicely fits in one of the major focus areas of Charité, which is developing therapies that involve T cells for treating cancer and possibly in the future, of course, also other diseases. So I would like to finish, just summarize that molecular imaging and theranostics are important building blocks of the translational value chain. We mainly focused on in vivo validation, also are now starting some early clinical trials. Of course, the goal would be to bring this really as a precision medicine approach into approval and show in phase 3 trials that these precision approaches using radio probes is a very valuable, novel, or I mean, I don't want to underappreciate that there are already that there is already a lot of use of such probes, but I think there is a lot more potential. So with that, I would like to finish. This is my current group in Berlin. I would like to thank the colleagues within the consortium here, my long-term colleagues at Innere Medizin 3, a lot of collaborations partners from here, TU München, Klinikum Rechts der Isar, and also some of my more recent partners who have contributed to the data I showed here. Thanks.
Thank you. Chemokines go their way. Time for one question. Ralf Weisler, unfortunately, is waiting. Or no, fortunately enough, he's waiting. But questions from the audience to this talk?
Where will you produce CA64 energy?
I think I cannot tell you because we don't have this possibility at Charité, so we will depend on our partner at Vivantes. It's really unfortunate. So das former städtische Kliniken Berlin.
One short question there.
Sorry, no imaging question, but just a brief biology question. When you do your cross your lymphoma model with this piggyback model and you find a synthetic lethal target synergizing with Mäcke, is this, in your experience, then typically lymphoma specific, or does it work in an overarching way in other entities as well?
It's a very important question. Thank you. We could show for SENP6, this sumo protease, that this protease, the gene, is also affected in uveal melanoma and other diseases in up to 15% of patients. And we are currently performing such experiments. So we think that this is a general mechanism and that we might be able to use it as a predictive biomarker, not only in B-cell lymphoma, but other diseases. So we already have some data from multiple myeloma. Again, it's a fraction of patients affected. So I would always aim for taking a look at other cancers too.
Thanks.
Okay, I think we have to finish, and I pass the microphone quickly to Markus to introduce Ralf Weisler.